So when you look at a biological channel, you look at a biological membrane, well, first you realize if I have a biological channel, it's sitting in a bilipid membrane. And that's, again, remember you're dealing with something that has a membrane of both molecules that like water, the circles here, and ones that don't like water. And so they stitch together very nicely. And so as a result, this has almost no conduction between it. It's a very tight seal. And so what you find is that this basically looks like a capacitance with ions sitting on both sides. It feels exactly like what you would expect electrically, except that maybe if you're an electrical engineer, you're not used to things being ions moving in a fluid. Fair enough. Then I look at this channel and I go, that's awesome. So I know I've got a capacitance. The channel allows ions to flow through. So ions moving through with different fields and so forth means I might be talking about some sort of conductance. Well, what's also interesting is because I've got separation of charge on both sides and they're different concentrations, that's naturally going to, across the semi-permeable membrane, create a voltage. That voltage is often called a Nernst potential. And it's just sort of a natural thing of integrating over a barrier where I have different charges on both sides and I sort of see how things equilibrate. Nernst potential is very similar to a built-in voltage in p-n junctions, and we can see this physics in many, many places, and the formulations are very similar. But I can imagine that. I can imagine conductance. Interesting enough, if you look closely, that conductance is going to be a function of what that membrane voltage, that inside voltage is. Why? Well, it turns out there's going to be some sort of connection to it, or some sort of sensing of the voltage, maybe sensing of the chemistry, and it's going to come back and affect it. Okay. So this is sort of the classic formulation that Hodgkin and Huxley developed in the 50s. Amazing sort of thing, Nobel Prize winning formulation, beautiful approach. The only thing is they didn't know anything about transistors at the time. Not shocking because this was just being formulated at about the same time. So no shock, no surprise that like double E's may not have been talking to neuroscientists. Even though there was a lot, I mean, it's still only so much to be talked about there. So the interesting part is you could say, well, what about instead of talking about a resistor, what if I talked about it as a transistor? And where this membrane voltage connects through this. And in fact, what's interesting is that people will talk about this as a channel and having charge going through the channel that is modulated by some gating function, which is really quite interesting because you would talk about the area of a MOSFET between these 2 points as a channel, and that terminal is a gate. And what actually happens if you look closely is that I really have a voltage control barrier here that goes between outside and inside. And so what I'm looking at here is if I look schematically, I'd have a membrane on both sides, and I have charge outside and inside, and charge can be equilibrating back through this. But if I looked across this membrane, I would basically be having a voltage controlled barrier. I'd have this sort of barrier with a channel and a channel potential that's being changed by some sort of gating mechanism, which is actually some interesting function that I can work with as I'm going through this. Really cool. What's even cooler is that if you look at a typical MOSFET transistor, particularly operating with subthreshold currents, is I can go from source to drain and there's a gating potential that's modulating the channel, again, that basically, if I were to look right along this edge and look at a voltage conduction along that edge, this is again a voltage control barrier. And so one of the things that's really important is to realize that looking at a MOSFET in this case really does model the physics quite well of what's going on through a channel. It certainly does not quite account for all the Nernst potential kind of questions, but the voltage control barrier is actually sort of the core concept that we have there. Now, granted, in biological structures, I've got some conformational changes that are going on, which is adding some additional 1/f noise and some other fun things to that. And there's some physics behind that, but I'm not going to bury you on that question here. Just to know that this equivalence is actually quite significant and actually quite valuable because I can then start pulling in intuition from different places to kind of think about whether it be circuits or biology, neuroscience, to kind of help and assist the other. So it's really pretty incredible what could be possible with these structures.